before we start, I'll repeat that for people joining late. Announcements on what is left about this week. So there are a few changes in the program and structure. So there are several people more than one people that have a problem on Friday because there is a conflict with the CISA data science entry exam. So the way we are going to deal with this is that we are going to move the exam of this course from Friday to Thursday. So the exam will be on Thursday. And since the exam is on Thursday, I shifted a bit, a few things that you were not aware of, so it doesn't change much. But it's a good time to tell you what is the plan for tomorrow and Wednesday and Friday, because we are going to have a lecture on Friday. So what are we going to do tomorrow? So you saw that the title of the course was Quantitative Physiology, which is this one, and Critical Thinking. So the way we are I shifted things around is the following. So today we are going to finish with the Blackboard lectures. Then tomorrow we are going to do something more hands-on. So when you are going to work, and I'm going to sit and watch you. No, I mean, I'm going to stay there. And so basically what we're going to do tomorrow is that we're going to go to the computer lab. So bring your laptop. And you are going to be divided in groups, randomly assorted by me with a very fancy criterion. And you're going to play with a data set that it's related to what we did these days. And so basically, I'm going to ask you very broad, undefined questions, and you're going to play with this data and try to answer them. Okay? And then basically, you're going to work in group, and your goal is to produce like a notebook or a code or some figures. And then this part here is going to be part of the exam. And we can say that the deadline is on Friday, so you have time to work for it. Okay? Then on Wednesday, I'm going to do a presentation, a lecture on things about, let's say, critical thinking in the area of big data. Which is not going to be part of the exam. So on Wednesday, you stay there and relax, stay there and sleep or whatever. But that is not going to be part of the exam. Then on Thursday, there is going to be the exam. And then on Friday, we can go finish if I didn't finish something on Wednesday, plus exam correction. We can see how it is. Okay? Is the plan clear? Good. Okay. Yes. Yeah, the exam is going to be exactly the exam, the end zone, and the end zone. It's like you're going to do that, and then you're going to submit the thing on when we say, and then the exam is going to be like a list of questions and problems. I mean, the criterion that Matteo gave us is if you didn't sleep during the lecture, you should be able to pass the exam. Okay. Okay, so it's like some problems, but yeah, I mean, you have to study, but it's not like something, I mean, take it with a good amount of stress, okay? Not more than that, not less than that. Questions about that? About the plan? Okay, so we can start with the point where we stopped on Friday. So there is the link I sent you to the notes. Some people, I mean, since I wrote it on the board last time, it was unclear. So this should point to the right URL. Now we can start from the point we stopped. And actually, I can answer one question that is related to what is happening in this case. So basically, we were studying the situation where cells are exposed to some amount of antibiotics. And our goal was to understand, well, we saw that the dependency of the growth rate on antibiotics has this weird shape where sometimes it increases, sometimes, I mean, where there is this effect of the nutrient quality, which sometimes modify this response to the concentration in different ways. But one question that was asked just at the beginning of this lecture was, what happens in presence of antibiotics? So we have these two sectors, the ribosomal sector and the P sector. How does a cell regulate these two sectors in presence of antibiotics? So now there are two ways to answer any question, and particularly this. And so the two ways are what happens empirically and how does a cell implement that. The difference is, in one case, it's a question about what is the, if you want, the fixed point of the internal dynamics of the internal concentration and regulation, and the other question is how does a cell do it. So the point is, what happens in absence of antibiotics is that you have like your Q sector, okay? And then you have something which is P and something which is R. Okay? Now, what if you add antibiotics? Now, without writing any equation, you can think about it with the example of the fishermen and the people cooking the fish. Right? So if you add antibiotics, what happens is, you can think it in a different way, but the outcome doesn't change, right? You can imagine that the example was there are people fishing, and then there are people like cutting the fish and cooking it. So you can think of antibiotics as something that makes the tools of the people cutting and cooking less efficient, or you can think at antibiotics, and this is more similar to what we have done, as something that takes some people that are cooking and sort of put handcuffs on them, so they are not able to work anymore. Okay? So in both cases, what happens is that what is going to happen is that you are going to be slower in the second process, so you are going to accumulate like fish to be processed. And so what you have to do is that you have to reallocate people from that are fishing to the second step. Okay? So what we expect to see is that you increase this R. Okay? And this is exactly what happens here. Okay? Now, how does a cell do it? This is the second way to answer this question. Is that in both cases, both if you imagine that is like something about the tools or something about sequestering, sort of removing people from the point, you have these two steps. You have the nutrients, the amino acids, and the proteins, right? The point is there is a feedback here, right? So the more things you have here, in the example, the more fishes you have accumulated, the more you want to produce ribosomes. So the more you want people to process them, okay? So the point is, if you make this more slowly, you're going to have an accumulation of these amino acids, of these building blocks. And so what a cell does is that it's going to produce more ribosomes as a response, okay? Until this balance, and so you have the two fluxes balanced, okay? Does it answer your question? Okay. Other questions about what we did last time? Okay. Good. So this was the sort of setting we started from. And just to recap, what we did was to write equation which describes these processes. So the fact that the amino acids enter the cell in a non-passive way, so there is active transport. And then, not amino acids, sorry, the antibiotics enters. And then the antibiotics can bind to the ribosomes. And so then only the unbound ribosomes can translate proteins. And then we had the other two inputs of our models, which are the two growth laws. So basically that the growth rate is equal to the concentration of unbound ribosomes times their elongation speed, and is this fact. And the second one, if you want, is this feedback mechanism, okay, which determines how many ribosomes we are producing. And this is, however complicated it is, we know that it has to follow, effectively at the end, it has to follow these empirical patterns. And so we put that input, and basically we have, of the we have 4 unknown variables and 4 equations. And the 4 unknown variables are the internal concentration of antibiotics, the number of unbound, the concentration of unbound ribosomes, the concentration of bound ribosomes, and the growth rate. We have these 4 quantities here. We have 4 equations. And so what we can do is that we can express the growth rate of a cell as a function of all these parameters and the external concentration of antibiotics. Okay? And so there was some algebra involved that you can find in the notes online. And at the end, we arrived to this expression here, okay, which we obtained. The only approximation we had to make was that K-on was much faster than kappa T. Okay? So we obtained this equation here. And this equation, it's an implicit equation. It's an equation that you have to solve to get what is the value of the growth rate as a function of all the parameters and the concentration of antibiotics. Okay? So by solving this equation, you get lambda over lambda naught as function of the external concentration, which is the plot we started from. Going back, is this plot here, right? So these plots here are exactly lambda over lambda naught as function of the concentration of antibiotics. Yes?
So lambda bar, all the parameters in lambda bar is down.
Yeah, I'm going to go to that. Okay? And so our goal is to have a plot here, and of course in this model we have parameters, which are parameters that specify how a cell interacts with antibiotics and the properties of the antibiotics. So the parameters of our model are kappa T, which is the translation speed, okay? How fast the ribosomes produce proteins. Then we have P in and P out, which represents these 2 arrows, right? How fast antibiotics enter the cells and go out. And then we have K on and K off, which represents the rate at which the antibiotics binds to the and unbinds to the ribosomes, so these 2 arrows. And then we have Rmax minus R naught, which represents, R naught is R mean, which represents this span. So if you want, it represent how much the amount of ribosomes can vary. Okay? So we have these parameters, and through our calculation, under this assumption, we found out that we can express all these parameters in terms of 2 parameters, one with the units of growth rate and one with the unit of antibiotic concentration. Okay? So this one as the unit of a growth rate, and this one as the unit of concentration of antibiotics. Okay? Questions about this? Okay. So now, this is a cubic equation. You can solve it numerically. And you can get an expression of lambda over lambda naught as a function of Ax, where there are 2 parameters you have to fit. Right? You have to fit lambda bar and A bar 50. So in principle, you have more parameters, right? You have 4 arrows here, plus 2, you have 6 parameters, but what you discover is that your expression depends just on 2 combinations. Okay? So what you can do is that you can fit these parameters to the profile. No. Okay? Now, so for each panel represents one drug. Okay? And here you have all the glycerol medium. So you have like 3 conditions where you have glycerol and just a simple salt, glycerol plus amino acids, and glycerol plus rich medium, right? So these were the plots we started from for the 4 drugs, and you have the growth rate as a function of the concentration. Okay? Now what you can do is that you have 2 parameters, okay? And these 2 parameters will depend on the drug. Okay? But the idea is that these 2 parameters do not depend on this condition. The only thing that changes here is lambda naught, which is known. It's not something we have to fit. Right? It's this lambda naught. Okay? And so each of this curve, each of this panel, you have 2 parameters for the full panel, right? Okay? So basically what you have 1 parameter that tells you what is the value, the typical value of the concentration where you start to see an effect. And the other one, it tells you something about the bending on this curve, okay? The sort of concavity of the curve, which is lambda bar. Okay? And what you see is that with 2 parameters per plot, you are able to see this difference, right? You already see that, for instance, in this case, you can predict, you can see that this goes up. In this case, you can see that this goes up as you increase the movement. Okay? Then you can repeat that for the glucose condition, and it also more or less works well, where you have the different conditions, okay, and different parameters. Questions? If you have a comment, you can say it. You want to say something? No, okay, good. So basically, you have a model that it's like simple enough because it has 2 parameters for all. If you fix a drug and you fix the sugar, you have 2 parameters and you can reproduce the dependencies across growth rates, across values of lambda naught, which is what we wanted. And in particular, what we can see is that it reproduces the fact that for some condition, as you grow faster, the curves are arranged in this order, while for other curves, they are arranged in that order. Okay? If you remember, I should have said that before, what we started from last time was this observation that sometimes, as you increase the growth rate, the curves are arranged in this order, and sometimes they are arranged in this order here. Okay? Good. Questions about this? Okay. So now what else you can do? Well, you have these parameters. The observation we wanted to explain and describe was, so you have these curves that does something, and when this is half, you have a value of the concentration of this inhibitory concentration where the growth rate is half of what is started from. And the observation that we had is that this A50 as a function of lambda naught sometimes goes up and sometimes goes down as a function of the type of antibiotics, right? As you can see here. So in this case, as you grow faster, this value goes up. In this case, as you grow faster, this value goes down. Okay? So we sort of did it quickly last time, but how do we find this value here of the inhibitory concentration, what we can do is that we can say, okay, A50 is the value of the external concentration when the growth rate is equal to lambda naught over 2, right, by definition. So if we substitute over there lambda equal to lambda naught over 2, here we get 1/2, which simplifies with the 2, and then we get, here we get lambda naught over 2, so 1/2 lambda naught over lambda bar, and here the 2 simplifies with this one, and we get plus lambda bar over lambda naught equal to A50 here, so we get that A50 over A bar 50 is equal to this quantity here, okay? Okay? And so if you plot this function, so this inhibitory concentration, which is what we were looking at here, for instance, okay? So this is not nice, let's look at another one. So basically we are plotting this quantity here as a function of the nutrient, the growth rate in absence of the drug. Okay, so A50 as a function of lambda naught. Okay, and this is our prediction. And so if you plot this function here, what you get is that this function has a minimum when lambda naught is equal to lambda bar, which is here, and in that value you get the value A bar 50, okay, and the function is something like this. Okay? So basically, you have, if you want, you have, well, the first thing is you have two parameters, lambda bar and A bar 50, and so lambda bar, it tells you the value of the growth rate, the nutrient, the growth rate in absence of the drug, where you have the lowest possible inhibitory concentration. And the value of the inhibitory concentration in that value is A bar 50. Okay? And these two are expressed as a function of the parameters. Okay? So now you see why I put the 2 here, is just because I wanted to have this nice interpretation of the result. Okay? The other interesting observation is that as a function of lambda naught, depending on whether lambda naught is lower than lambda bar or larger than lambda bar, you can have that this function is decreasing or this function is increasing. Right? And so now what you can do is that you can say, okay, but now I have, I fitted all these curves, so for each of the drug and for each of the nutrient, I know, sorry, for each of the two types of nutrient, glucose and glycerol, I know what is the value of lambda bar, and I know what is the value of A bar 50. Okay? So what I can do is that I can say, if I plot A50 over A bar 50, which I fitted, as a function of lambda naught, which I measure, over lambda bar, I should get a unique curve, which looks like this. Okay? And so you can fit all the parameters, and this is what you get. Okay? So you get the 4 drugs. And you get the curve. And you get that the data collapse on the curve. Okay? And so you see that you have some drugs where this goes down, some drugs that this goes up, but they sort of fall within the same phenomenon, the same phenomenology. Okay? Questions about this?
So you said this diagram is equal for all the
Yeah, so what you have to do is that you say, so there are 4 drugs, okay? And the other observation, which I didn't emphasize, but I can emphasize, is that when we had 6 nutrient condition, and the observation for the nutrient condition is that there is a difference. So you have, let's say, in principle, you have you have the different drugs, and you see that these parameters here are drug dependent, right? For instance, K on and K off, you expect them to be drug dependent. Right? P in and P out, you expect them to be drug dependent. Okay? So these 2 parameters will be drug dependent. The other observation you make is that, and you could see it from here. Well, let's look at it from, doesn't matter. So that you have these 6 conditions, right? Glycerol, glycerol plus amino acids, glycerol plus rich medium, and you had glucose, glucose plus amino acids, glucose plus rich medium. The parameters, right, these 2 conditions, so glucose plus something or glycerol plus something, corresponds to different parameters. The reason is that, for instance, the kind of transporters that they express, so the way the antibiotics enters the cell or is sort of transported out, depends on whether you go on glucose or not, or on glycerol. Okay, so you have also this distinction here, which I didn't emphasize. So basically, the point is, you fix the sugar, let's say glucose, you fix the drug, let's say chloramphenicol, you feed these 2 values, right? And these 2 values, you can feed them to any of these 3 conditions, right? You fix these 2 parameters, right? And the same for the others. And the point is that what you get for free is that you then rescale the growth rate by these fitted parameters, and you get the collapse. Questions? Yeah. Lambda 0 star is lambda bar. Sorry. Yes. It's what we call the lambda bar. And these IC50 and IC50 star is what we call the A50 and A50 bar. So it's exactly this plot here. Okay. Yes?
What are the difference between this, the plot, the squares and the
I think they corresponds to glucose versus glycerol, if I remember correctly. So you can, you have multiple, some of these points, so each point corresponds to 1 nutrient condition, right? So you have, in fact, 3 and 3, right? Yes. So you have nutrient conditions where is glycerol plus something or glucose plus something, okay? Square, I think, are, I don't know, because they are scaled by lambda bar, so it's not obvious, yes.
And these are 4 experiments, each experiment with a drug.
These are many more experiments. These are 6 times 4, it's 24. Because each point, well, no, actually, it's many more experiments, because you have, for each drug, let's say chloramphenicol, you have to choose 1 nutrient, let's say glucose, and then you have 1, 2, 3, 4, 5 points. Sometimes 6. No, 6 points. 6 points. So it's 6 times 6. So 6 values of the concentration times 6 nutrients times 4 drugs. So 36 times 4. 132. No, 138. 144, whatever it is. Okay? And for each of these conditions, what you do is you make sure that cells are growing exponentially and you measure the growth rate. Right? Questions about this?
Sorry.
Yes.
Just to understand, so we're getting
greater than 1.
Yes, so basically what you discover is that there is this value here, lambda bar, okay, which is a function of the parameters, okay? And basically it's something that expresses how fast, the antibiotics binds and unbinds, okay, this ratio. How fast is sort of pushed out, and how fast ribosomes elongate, okay? And if the growth rate is larger than this value, the inhibitory concentration increases with the growth rate. If it is below this value, the inhibitory concentration decreases, okay? Of the growth rate? So these two values here, A bar 50 and lambda bar, which are the two values that determines basically what is the value of the inhibitory concentration here, and what is the scale at which, of the growth rate at which you have the two phenomenologies, the two regimes. These are a function of the parameters of how that specific antibiotics interact with the cell, okay? And what is basically the range of ribosomes you can, and the properties of ribosomes, okay? Other questions? Yes. Because you have a wide range of variability of these things, right?
So it's a natural logarithm because it's 1 to the base 10.
No, but these are logarithmic, but yeah, I mean, if this is in log scale, it doesn't matter. I mean, what it matters here is that here you have 1, here you have 10, right? This is the value of this quantity here. The scales are logarithmic, but here it's equal to 1, here it's equal to 10. Okay. Other questions? Okay. So how can you further test this model here? Another way to further test this model here will be to try to modify something in the model. Okay? Now, how do you modify something in the model? Well, of all the parameters, right, which one is the one which is easiest to modify or easiest to test the effect? Or one you could think about testing the effect? So think about the parameters that appear inside the definition of lambda bar and A bar 50. There is one we already saw. KT. So if you take a mutant, you can take a mutant with the different values of KT. Right? With kappa T. So a mutant that has ribosomes going at a slower speed. Okay? So what you can take is to take a mutant, right? This is a plot without antibiotics. You can take a mutant that has ribosomes that go slower. Right? And so if you have ribosomes that go slower, what you have is that when you do the plot of phi as a function of lambda naught, what you expect that if this was the wild type, the mutant is going slower. So it has a slope. The slope of this is 1 over kappa t. It's exactly 1 over kappa t. So these curves are lambda naught over kappa t. Okay? So if you have a lower kappa t, you have a higher slope. Okay? And so basically what you can do is that you can, if you risk, I mean, you can test with this, but if you look at the value of the, this kappa t produce an effect on this lambda naught, okay? And there's an effect on the value of A50 at any given value, okay? So what you can predict is what is the value of the ratio of A50 of the wild type and the mutant as a function of the ratio of lambda naught of the mutant over lambda naught of the wild type. Okay? So I didn't derive this prediction, but basically you can imagine that you can, this effect kappa t will affect lambda bar, will affect A50, okay? Will affect lambda naught in a predictable way because it will be always about the same parameter. And then you can look at what is the value of A50 for the mutant over the one of the wild type as a function of the growth rate of the mutant over the growth rate of the wild type. And what you expect is that these two are related across the different nutrients, and the slope of this relation is this ratio here, which is a constant. Okay? And you get that this prediction holds. Okay? Which is highly non-trivial, right? Because what we did here is that we took a different bug, right? And we repeated basically the growth trajectory in the presence of antibiotics, and we expect to see a different value of the inhibitory concentration, okay, and a different value of the growth rate, and we can predict what is the relation between the inhibitory concentration and the growth rate as a function of one unique parameter, which is the translation speed, okay, the difference in translation speed. So this is really a test that the model does what it expected to do. Right? It's a pure prediction. Is it clear? I mean, what is crazy if you think about this plot here is that here you do something in absence of drugs. You fit what is the value of kappa t of the mutant, and then this is a new experiment, pure prediction, nothing to fit. Okay. Questions? Yes.
Can we further test on producing the useless proteins at before?
Yes, that could be a good idea. They didn't do it, but you could do it. No, I mean, you could in theory, right? Could think about doing it. Yeah. And you can generate a prediction of what will happen there.
Would it be useful, for example, in this case, it would affect Rmax, which only appears in A50?
Exactly.
Would it be?
Yes, it affects. Yes, but it would also affect lambda naught. Right? So it depends what plot you do, but the point is that it will affect lambda naught without affecting lambda bar. Right? So it affects what is the value of A bar 50. So it moves this up, but you also move this lambda naught here, right? So it's not just, I mean, you could say, yes, I mean, it scales this value, but if you fix the nutrient, it's not just scaling by this value here. Questions about this? Sorry? I didn't. No, this is the mutant. Mean, it's just because it's the name of the mutant, I think. It's just because it has a lower translation. It's just the name of the mutant. Okay? Good. Okay. So what I want to do in the second and the last part of the lecture is to sort of further extend this and try to see to use this framework to understand something about evolution. Okay, so it's something even more pushing it to the extremes. Now, the point is, as I told you, resistance evolves also in the lab, right? So the idea is, if you expose some bugs to the drugs and you wait, you have them growing in the lab, they will evolve some mechanisms to improve their growth in presence of antibiotics. Okay? So how can you do that? Suppose that you want to evolve a resistance in the lab. How do you do that? Well, one simple way to do it is to do like an experiment where you keep growing bacteria in the same condition in presence of some antibiotics. So for instance, what you could do is that you have your bacteria, you have some antibiotics and some nutrients, they grow, they go lower, right? They go lower than in absence of antibiotics, of course. Then you take like a small sample and you use it to renoculate in presence of antibiotics to fresh medium, and then they grow. And then you take these and you renoculate, and then they grow, etc. Right? So you keep exposing them to the same environment where you have fresh medium and antibiotics. And so basically you can start to have mutations, okay? And they evolve some level of resistance. Okay? So empirically, suppose that you start from the curves we saw, and you look at the wild type, so the original one, you have like some dependency of the growth as a function of the antibiotic concentration. Right? And then you take one of these mutant, okay? And let's say that one of these mutant was evolved in a condition where there was some level of antibiotics. Okay? So let's say that the level of antibiotics was, I don't know, here. Okay? Was somewhere here. Here we are comparing the curve for the wild type with the curve for the mutant. Okay? So let's say that this was grown at 2 microgram per milliliter concentration of antibiotics. What you see at that concentration, the mutant grows faster than the wild type. Okay? Because the blue line is well above the gray line. Okay? Is this clear? Now, what you saw is that now you take this mutant and you repeat, you have the mutant and you grow them that at different concentration. What you see is that it grows better at higher concentration of the antibiotics, but it grows worse than the wild type at low concentration of antibiotics. Okay? Is this plot clear? Some say yes, some have doubts. If you have doubts, ask questions, because many others might have doubts.
Why is it not doing as well as the wild type in the low concentration?
We're going to ask that. But the important is you understand the phenomenology. Then the why, this is the rest of...
These are very weird things that like you keep saying the last session and this session. Like, it's weird. Why, if it's just like, if you just think about it, I know it's not like obvious, but if you think about it, it's just resistance. So like,
Yes.
antibiotics shouldn't affect in...
But basically what we're... So there are... Okay. So your questions, there are 2 types of questions. Okay. One question is why this is happening, and then is what the lectures is about. So I'm not going to answer now. Okay. The other question is you understand the phenomenology. So what I want you first is now to understand what this plot means. then if you understand what this plot means, you should be surprised, or you should say, why, okay? But I want that you understand why you should say why, okay? Is this clear what this plot means? Yes.
Can I ask you, how do you rescale the number?
So this is all rescaled by the growth rate of the wild type in absence of antibiotics. So the one here, so the rescaling here is to the same value, okay? So let's say that the wild type was growing at 0.4 duplication per hour, okay? You rescale the growth rate by that value. In absence of antibiotics, this is growing at 0.6 times 0.4, okay? So they are comparable, right? Yes? The x-axis is the external concentration of antibiotics, the antibiotic concentration. Okay? So this is one mutant. You can repeat it for many experiments, and I'm going to show many experiments, and what differs between these experiments is that these experiments were performed at different concentration of antibiotics, right? So you can expose the bugs to different concentration of antibiotics. Why it's always okay? There is some bug in my slides. Okay. So what you see here is multiple evolution experiments. And what you always see is that the curve of the mutant has always the same phenomenology, okay? What varies is that sometimes it's closer to the wild type, sometimes it's very distant, okay? And the difference is the value of the concentration of the antibiotics they were exposed to, okay? Now, the observation which is in common to all these curves is exactly related to the question that was asked. What you see is that at the, let's say, at the value when there is high enough antibiotics, the mutant grow faster. But sometimes the fact that evolved the ability to grow in presence of antibiotics implied that it was less good at growing in absence of antibiotics. Okay? So that there is a benefit when you evolve the ability to grow in presence of antibiotics, somehow you grow worse in absence of it. Okay? So there is a cost, a trade-off. We're going to go to answer that. I'm glad you are asking. Okay? Okay, this is exactly what we're going to answer. Okay? Okay. So the question we're going to ask is, yeah.
So the experiments were done like you said before. So these are the plots coming from
The end.
the resistance.
Yes, so basically you repeat these many times. So you keep passaging these many times. Okay? I don't remember what it was, but for this resistance it's probably on the order of weeks. No, no, you have one line, and one line refers to one of these mutants. So you repeat this experiment for, let's say, 3 weeks. Okay? Every day you take it and you expose to the same medium. So always the same concentration of antibiotics. Okay? Always to, I don't know, 2 micrograms per milliliter. Okay? Always the same for 2 weeks. At the end, you're going to have a bug that is different from the bug at the beginning. It accumulated mutations. Okay? And then you take this bug, and then you grow in all these conditions, and you look how fast it's growing. Okay? Is it clear? No. Yes? No. It's not clear? So imagine that you repeat this every day, right? And you you just take, so it's like the baking yeast you have. So every day you add flour. You take a sample, you add flour, and you have it growing, right? In this case, every day you take a sample, you put it with new food and some antibiotics. And you do every day for 3 weeks. At the end of 3 weeks, what you do is that you take a sample of these bugs, and you look how they are growing at different concentration of antibiotics. And what you see is that they're growing differently. If you measure the growth rate, their growth rate is different from what it was at the beginning of the experiment. This is the observation, okay? Why is it different? Because they mutated, the accumulated mutations, they evolved, okay? Yes?
Can you differentiate between the mutations?
Yes. I mean, for now, each plot is one line, right? You do one experiment. You do another experiment. I mean, for reference, there is one experiment which is called the long-term evolution experiment, which has been going like this every day since 1987. Okay? So you can look at it online, what they found. I mean, you can imagine that you can repeat every day for more than 30 years. Okay? This is an absence of antibiotics, very simple medium. But this is a standard, yeah.
Excuse me. If we could take an example, for example, the third line, the last graph. And on patient 4, there happens something, goes down until it disappears.
Yeah, you see that, I mean, the curves have a similar shape than what they had before. I mean, these curves, as you expect, are fit of this model here. But the point is, what I care about is that this mutant was evolved at some concentration, let's say at 3, okay? And then what you do is, at the end, so you expose this bug, you take the wild type, you expose the wild type for 3 weeks at a concentration of 3 microgram per milliliter, concentration of antibiotics, and at the end, you measure again how they grow, and you see this curve.
So before it disappears.
Yeah, but before it was not growing at 2. Okay?
So
Yeah.
it only takes 3 weeks to...
I don't remember if it was 3 weeks, but it's
No,
reasonable,
I mean,
yes.
It's a short time. Like,
It's a short time, they're fast in evolving, yes.
so you can have like multiple mutations.
Yes, so you can ask about where the mutations are, okay? You can ask where the mutations are, and so basically this is like what you can do is that you take these different mutants, you sequence them, and you compare their DNA sequence to the DNA sequence of the wild type, and you can see where are differences, okay? And if you do that, what you find is that you have differences typically appearing in genes that are related to the membrane, okay? So to the transport of the antibiotics. So the way it seems that they evolve is something that is related to the membrane. Yes.
I mean that in the last like 20 years, the environment changed a lot. So the bacteria now is in nature. It will be different from...
They are always evolving.
yeah.
Yeah. Evolution always happens. I mean, everything is always... I mean, here you exposed just this experiment here that has been going for 30 years. So 30 years for bacteria, it's much more than the time it separates us from the Neanderthal, okay? And they are in a very simple condition, always the same, they are still evolving. So there is very much room for evolution. The evolution is at the same time very fast and very long.
This is dangerous for us because they will have resistance to all the antibiotics.
No, here you are exposing to one type of antibiotics.
No, I mean in nature. In nature, because...
In nature, there are yes. So in nature, yes. The point is, in nature, there are bugs that are resistant to all the antibiotics. The point is that resistance comes with a cost, right? So you might be optimal for growth at a given concentration, but then you do very bad when there are no antibiotics, okay? So not all bugs are resistant to all antibiotics, because you become a resistance, you have to pay a cost.
There is no super mutation.
No. No. I mean, but the point is that if you expose them to an environment where they are always exposed to antibiotics, then being resistant to antibiotics is advantageous, so there are going to be many bugs which are resistant. Yes? There is.
So if you have an infection with a bacteria that is resistant to some antibiotics, it's better to have take a low concentration.
No, I mean, no, it's much more complicated. I mean, actually, if you take the worst thing you can do, which increase the speed of evolution, is if you take a low dose of antibiotics and then you slowly increase it. Right? Because if you do that, you have a small effect on growth, so they grow faster. They still have an advantage, so they are able to evolve. And then basically you are paving the way to evolve high resistance. Right? Imagine that if you have the wild type, right? If you expose them at this concentration, it's going to be very easy for them to evolve resistance. If you expose them at this concentration, they are not growing. Right? So there is no room for evolution. Okay. So the point is, I mean, one can study what are the mechanisms, one can sequence. On one end, it's complex. There are a lot of things here that matters. The common message, what we care is that it's something related to membrane and transport. Okay? Good. So how do we understand this? So, okay, we want to characterize and understand basically what is the space for bugs to evolve. Now, what does it mean that they evolve? It means that they change something about something in their genome, and so this will affect their properties. Now, in our world, right, in our modeling setting, the fact that they change their properties, it means that they change the value of some parameters here. Okay? So the question is, what are the values, which parameters they are changing? Okay? Now, again, we are in one situation where in principle many things could happen. And so I'll tell you many things could happen. So one could think at a very complicated model where everything can change. The point is that the message that I want to give you is simple. And the main parameters that is actually changing, I mean, the first observation that we have is that most of the mutations of this mutant are related to the membrane. Okay? So if they are related to the membrane, what do you think are the parameters that are going to change? Yes, so P in and P out. Actually, P out is something that is not really related to the permeability of the membrane, it's related to active transport from inside. So actually the main parameter is P in. Okay? So do you think it's better to have an higher P in or a lower P in? Lower P in. So what we're going to say is that P in of a mutant, okay, is going to be epsilon times P in of the wild type. Okay? Where this epsilon is a parameter that tells you what is, well, the rescaling factor. Okay? Now, we don't know what is the value of epsilon. The value of epsilon will depend on the particular mutant, the concentration they evolved in, but it evolved at. Okay? So this is what gives advantage to the mutant. Now, what we see is that the mutant also pays a cost. Right? If you take a mutant evolved with a given concentration of antibiotics and you exposed it to not to antibiotics, you're going to see a slower growth. Okay? So we must have another parameter that changes. What is the other parameter that changes? What do you think it's related to? I mean, how can another parameter change it? It means that if I change something in the membrane, right? I change something in the transport, for instance. Another parameter is also related to the transport, right? If I take more antibiotics inside, I will take more or less of something else. Right? That the two are related. What do you think is that something else? Nutrients, right? What you're taking from outside is, well, in this case, antibiotics and nutrients. Right? So what is the parameters that sort of tells us something about nutrients taken from inside, from outside to inside? Kn, yeah, it's a tricky question because it's the only parameter that's not appearing here. It's Kn, right? So the idea is that we have Kn, which is the rate at which we process like nutrients to amino acids, sort of these corresponding parameters. The idea is that the Kn of the mutant is going to be proportional to Kn of the wild type, and what we can say is that it's the same proportionality, right? So somehow you have a transport of the nutrients, and the idea is that the transport of the nutrients and the transport of the antibiotics are through the same channels, which actually in many cases is true. So if you evolved the ability, if you transport inside less antibiotics, you also take inside less nutrients. Okay? So if you take inside less nutrients, you're going to go slower, right? So this is what gives the cost. Okay? So now, where in all these parameters here, where does Kn enters? In lambda naught, right? Kn enters in the growth rate in absence of antibiotics. Okay? So in particular, if we remember the things we brought, lambda naught will be, well, R max minus R0, Kn, Kt, Kn plus Kt. So you have that 1 over lambda naught is equal to 1 over R max minus R naught, 1 over Kn, 1 over kappa t plus 1 over Kn. Okay? So if we introduce the fact that Kn is equal to this one, what we have is that the growth rate of a mutant, a bug, okay, will be equal to, well, here it's epsilon Kn of the wild type. Okay? And so if you expressed Kn of the wild type with the growth, so you also know that one of so you can express the growth rate of the mutant as a function of the growth rate of the wild type, right? You invert this relation, you find Kn of the wild type as a function of lambda naught of the wild type, you substitute it there, okay? And what you get is that lambda naught of the mutant over lambda naught of the wild type, which I'll call basically the cost, let's call it W of epsilon. This is a function of epsilon, right? It's epsilon over 1 minus epsilon. And here you have lambda naught of the wild type over what is lambda max. And lambda max is simply R max minus R naught times kappa T. Okay? So what you see here is that this function here, it's an increasing function of epsilon. Right? So it's an increasing function of epsilon, right? So if you have, no, sorry, I said something wrong. Yes, it's an increasing function. Sorry, I was very confused. It's an increasing function of epsilon, right? Because if epsilon is equal to 1, it means that you are exactly equal to the wild type, right? It's how much you transport relative to the wild type, okay? So if this becomes equal to 1, you get exactly 1, so you grow as the wild type. If this is lower, you go less than the wild type in absence of drugs, okay? So this is to say, if you take one of these mutants and you don't have drugs, right, you're going to go slower in absence of drugs as a function of this epsilon. Okay? So this is better, increasing epsilon in this respect. Yes?
Can we say that epsilon is a quantitative way of looking at different mutations?
Yeah, we can say the mutation, what affects, yes, what characterizes different mutations is that they have different values of epsilon. And then the values of epsilon modifies the value of Pn and the values of Kn.
And why is the epsilon in both?
Because the idea is that both the nutrient and the antibiotics enter through some active transport, and they are affected in the same way.
So like the main thing in the mutants is that they have different membranes?
Yes, there are mutations that are related to membrane, yes. Okay? Good. So the other effect that we have to calculate, which is a little bit more, well, it's not more tricky, but it requires a little bit more analysis, is what is the effect of the mutant, of the mutations on the response to the antibiotics. Okay? So how do we quantify it? Well, the first point is that we see that the lambda bar doesn't change. Right? The only thing that changes is that A bar 50. Okay? So A bar 50, what we see is that A bar 50 is going to be equal to 1 over epsilon of A bar 50 of the wild type. Okay? Now, what we can do is that we can ask, okay, we can calculate what is A50, which is equal to A bar 50 over 2 lambda naught over lambda bar plus lambda bar over lambda naught. We calculated it before, right? So this is the inhibitory concentration, okay? And what we can calculate is what is A50 over A bar 50, A50 of the wild type, and we call it resistance, okay? And this is equal to the ratio of the A bar 50, so this gives an epsilon. And then there are terms that depends on lambda naught and the lambda naught of the wild type, okay? And so one gets an expression of, this is 1 over epsilon. One gets an expression which is a function of this ratio here, and it looks like 1 over epsilon W epsilon times R squared W epsilon squared plus 1 over R squared plus 1, where R is equal to lambda naught of the wild type over lambda bar. Okay? So you do some algebra, it's all in the notes. It's not very important now, the calculations. But what you get is an expression that tells you what is the effect on the inhibitory concentration as a function of epsilon. Okay? And the parameters that enters here is, well, this ratio here, and where you are as a function of lambda bar. Okay? Which is unaffected by the mutations. So at the end, what you get is you can take different mutants, okay? And what you can do is that for each mutant, you can fit what is the new value of A50, of A bar 50. So you can infer epsilon, okay? Yes?
The number bar was unchanged due to this. Number
Yes, because it depends on parameters that we assume they didn't change.
And so this R is independent of any kind of changes here.
It depends on epsilon. This R of epsilon is a function. No, yes, this is independent. This is independent of epsilon, and this ratio is independent of epsilon. Okay. So basically, this ratio here it tells how good is the condition with respect to the best possible condition for the wild type, and this tells you how good is the condition with respect to this scale of the antibiotics. And so you fit these curves, you get epsilon. What you find is that, well, you can reproduce this resistance, which is the value of the inhibitory concentration as a function of the inferred epsilon, and the growth in absence of the drug as a function of this epsilon that you fitted from the data. Okay? So this is just, these plots tells you that the model is good enough to be able to reproduce the data. Right? It's not really a test. And basically you get that the growth rate, that these two parameters are anti-correlated or like negatively correlated. So if you get, this is the trade-off we were talking about. Why you have this? Because if you have an higher resistance, okay, in order to have an higher resistance, you need to decrease the uptake rate, right? And so decreasing the uptake rate decreases the drug-free growth rate. Okay? So basically you can move here, right? Okay? And now you can say something else, as always. What you can do is that you can say, okay, but at the end, what I did was to perform an evolution experiment. I mean, this tells me that I have these different mutants evolved with different level of nutrient concentration, and they evolve different values of epsilon, and the values of epsilon stay on this trade-off line. But at the end, I did experiments with a given value of the concentration. Okay? So I have experiments with a given value of concentration, and this corresponds to a given value of lambda. Okay? But lambda, the growth rate of that bug with that concentration, is a function of epsilon. Okay? So if you want, what I have is, I think I can safely remove this part. No. At the end, what I have is, if I take this curve here, right, what I can write is that there is 1 lambda of epsilon AX, which is a solution of this line here, right? So if I fix epsilon, I can always solve this equation, and I get a different solution for different values of epsilon, right? Because different values of epsilon will change this A bar and will change the lambda 0. Okay? And then what I can ask is, for a given AX, what is the maximum what is the epsilon which maximizes the growth rate? Right? How do I do this? I basically want to find the values where this derivative is equal to 0. I take this derivative, I substitute it to this equation, and I get another equation for the dependency on epsilon. Okay? Yes?
How do you fix epsilon by taking just one mutant?
Yes, you take one mutant, but the point is now we want to ask, this mutant evolved in a given condition with a given concentration of antibiotics. Okay? And I want to know from the model, for that concentration of antibiotics, what is the optimal epsilon? What is the epsilon that maximizes the growth for that particular level of concentration? Imagine that I don't give any antibiotics. What is the optimal epsilon? 1. If there is no antibiotics, I don't care if this is equal to 0, lambda is equal to lambda naught. What maximizes lambda naught? Epsilon equal to 1. If now I increase antibiotics, well, I want to pay, perhaps, I want to pay some cost, decrease a bit the uptake rate, right? Because that will give me some resistance. Okay? And I can ask, what is the optimal choice? Okay? It's not obvious that this model gives an optimal choice. It could be that actually the cost, the advantage of resistance is always negligible, or the cost will be always negligible. I mean, both could happen. What you find is that actually, no, there is an optimal one. So then there is some epsilon optimum, which is the one that maximizes. And this epsilon optimal is a function of the external concentration, right? Because it's what I obtained here by maximize this by maximize lambda for a given external concentration. Okay? So what I get is an optimal value of epsilon for a given external concentration and the growth rate evaluated in this optimal value. Which is a function of the external concentration. Okay? And so if you do that, now again, this has no free parameters because I fixed everything. If you do that, what you find is that this predicts what happens in the evolutionary experiment. So here you have the growth rate. Okay? So the value, this value here, rescaled by lambda naught of the wild type, as a function of AX. Okay? The level of the concentration, well, divided by A bar 50 of the wild type, which is known. So it's the level of the concentration. And what you find is that the maximum growth you achieve in the experiments reproduces well what we predicted. Okay? Actually, what you expect is that this is an upper bound, right, for the model. Okay? And what you see in the color point are the strains that evolved in that condition, and the gray points are all the mutants that evolved in a different condition plotted in that concentration. Okay? So what you expect is exactly that the color point stay on this plateau, and the gray point stay below. And this is what you get. And this point is, if you want the relay, you can calculate what is the value of this quantity here at the corresponding values of the optimal epsilon as a function of the concentration. And again, you get a prediction, and this is what you observe. Okay? Yes?
So we're not claiming that every time we find a mutation that solves the stationarity condition. We say that we find whatever, and sometimes it will be the best.
I mean, if you look at the color point, it's very close to the best. Look at the color point. Forget the gray. I'll tell you, because the gray, it's a different thing, and they went very fast. So the color points is what you expect to stay on the blue line. Okay? So you evolved this bug in this condition, and this bug is growing in absence of antibiotics 40% as fast as the wild type, and this is what you predicted for these values of the concentration. Okay? If you were at these values of the concentration, you predict slightly below 80%, and this is what you get. Okay? The gray points is you take this bug, evolved in this condition, and now you look at the growth in this concentration, okay? So then it's suboptimal for that concentration, so it should stay below, okay?
Again, what's the gray point?
Huh?
The gray point. Can you repeat what is the
The gray point is you take this mutant, which evolved at a drug level equal to 3, and then you ask how fast is growing at a concentration 0.5. Now this mutant here is an epsilon, which is optimal for 3, but is suboptimal for 0.5. So it should grow less than the optimal level. And it does. Okay? Yes?
But I don't understand like what is the stochasticity of the evolution. It looks like
The stochasticity of the evolution, this is cool, it's a cool question. The stochasticity of evolution comes into the fact that the way you evolve these and the speed you evolve these, the trajectories that you take, the mutations you take are different. Okay? The time it takes to evolve these is different. But the point is, you see that and you have, you have some noise here, but what you see is that you have a sort of line you expect you cannot cross, and you don't cross it. Right? So it's like to say, I have a fitness landscape, right? And stochasticity enters in the way I explore this fitness landscape. Here I'm plotting how is the maximum values of fitness you can have. Right? So I expect the stochasticity change things, change the way I explore it, change how fast I arrive to that point, but it doesn't allow me to go beyond. Okay? Other questions? Okay. Now, I went fast on the calculations, but they are like not particularly convoluted, and it's everything on the notes. So, good. Okay, so let's conclude. So, other questions about these evolution experiments? Okay, so on this, what we found is that you start from this simple but rich physiology understanding of what happens in presence of antibiotics. And then you say, now I have evolution. Evolution means that some parameters have to change. You sort of get informed by the kind of mutations you see, and you postulate that what is changing is the rate at which you import antibiotics, and that is also related to the rate at which you import nutrients. And so using these, you can, at the end, you can predict what is the optimal value, how fast you can grow, how fast a mutant can grow in presence of antibiotics. Right? How much you can mutate and how much you can grow. Right? Good. Now, we took a sort of trajectory in these kind of models of physiology where we have these different sectors. One can use them, and I showed you like the basic setting and a couple of examples with antibiotics. One can use the same framework to explain many things about how cells decide to grow in different conditions. Okay? And so this is like a list of examples that then in the notes for each of these examples there are references. Okay? So you can use these frameworks to predict how much time does it take to adapt, physiologically adapt, so to start growing again when you change condition. You can use this framework to explicitly include like the previous steps, so transcription, DNA transcription, and the dynamics of messenger RNA. You can use these to model overflow metabolism, so basically when cells are growing, sometimes they secrete metabolites which can be used for growth. So for instance, bacteria growing in glucose, they can secrete acetate, which then they can use to grow. So they basically grow in glucose, poop acetate, and then they grow on acetate after they grow in glucose. And so the question is how much acetate they secrete as a function of their growth conditions, and you can use this to predict that. You can use this framework to study the effect of temperature on growth, which mainly enters in this kappa T, but also in the fact that you need another sector to deal with the stress effects of temperature. You can use this to study. So we always consider a single nutrient at a time. So how fast you grow in glucose, how fast you grow in glycerol, how fast you grow in succinate, etc. You can say, suppose that I have two different nutrients at the same time, and the bacteria grow on both of them. How fast does it grow on both of them, given that I know how fast it grows on one? It's not too high, it's not the sum, it's not the average. It's something more complicated that you can predict with these kind of frameworks. You can also use these, and this is actually something that is very interesting nowadays, applications of this framework to ecology. So there are many things you can read. If you look on the notes, there are references for this. And there are many things I didn't mention that probably would be worth to mention, but I forgot. I don't know why this is appearing. Okay, so this is the point where I started, and what I wanted to convince you of is that if you look at bacteria, even if we think that they're simple, they do interesting stuff, and there are interesting problems. So I was very pleased today that when I showed the plot, people were asking why. Okay, it means that they do that what you observe, the observations are surprising. The point is, even if it's biology, it seems way more, I don't know, intrinsically unexplainable. There exist quantitative patterns, which are not just a cloud of points with a line in between, but are really something akin to laws where you really see that what they can do, it's really constrained. What these biological systems can do, it's really constrained. And this allows to build really quantitative models that are not just a description of what you see, but they're really quantitative frameworks where you can derive predictions. Right? We saw it today. We say, suppose that you do a new experiment where you have a different KT, you can predict what happens. Suppose that you evolve the bugs, you can predict what happens, right? And when you predict, it's not just predicting, oh, this will go up, this will go down. It's really predicting the number, okay? So I hope that these were convincing example. And that's the end of the lectures on physiology, okay? Good. So as I said at the beginning, I repeat it now. So we have change of plans for the week. So I repeat everything from the beginning. Yes, questions? Sorry. Oh, yes. Thank you. Most important thing. I completely forgot. It's here. So please sign this. Questions?
What is the limitation?
Huh?
Is there a limitation to this framework?
No, there are limitations that, well, I mean, there are like important hypotheses this is based on. I think that the main limitation is that these are experiments with E. coli, with a particular bug, that is something that is representative of many bugs, but is like something also with many specificities. Something that has been growing for a long time in the lab. It's a relatively fast grower. Many of the things outside are not there. Another limitation is that these are experiments in the vacuum, so at balanced exponential growth. Many of the phenomena that happens are not in balanced exponential growth. I think it's not really a limitation. I mean, it's like, but it's not obvious how to take this and to translate it to something real. But this is, yeah. If you throw a rock from the Tower of Pisa, it's not obvious that you're going to explain stuff sinking in the sea. Right. Yeah. Other questions? Okay, so plans for the next days. So main shift of plans. So since people have the exam of CISA on Friday, the exam for this course will be on Thursday. So the exam will be on Thursday morning. Then shifting around things. Tomorrow, we're going to do an hands-on session on some data that I will explain tomorrow in the computer lab. So bring your laptop. Okay? You're going to be divided in groups. You're going to play with data. And then what you have to produce is to produce a notebook with some analysis that will be part of the exam. Okay? You're going to work in group. You will have a deadline which can be like end of the weekend. So you have time. You can go beyond the data sciences entrance exam if you care about it. This is going to be tomorrow. Then on Wednesday, I will do a presentation here on critical thinking, big data, blah, blah, blah. Something that you will not need for the exam. So you can sit and relax, sit and sleep. And then on Thursday, there is going to be the exam, which is going to be like problems related to physiology. And the criterion is if you didn't sleep during the lectures, you should be able to pass, right? This is the official criterion. So study, repeat the material, but don't be overstressed by it. Then on Friday, if people have the entrance exam of data science, they do it. We are going to do here, I mean, if we didn't finish something on Wednesday, we're going to finish it. And then we can have some questions, correction of the exam. Questions about the plan? Good. Yes? It's like, let's say, simple problems based on this. Okay. So it's just to see if you got the basis of the, important. For the exam, you can bring all the material you want, okay? So your notes, you can bring whatever. You can bring also in the tablet, yes. Okay? I don't know if this should be something you like or something you don't like. Because if you can do it, it means that you don't need it, right? Okay. Okay? No, but you can use also internet, whatever. You can ask ChatGPT to answer for you. Okay? I mean, it's real life, right? In real life, you can do whatever. Good. Okay, see you tomorrow. I'm serious, yes.